I'm going to go through modeling meiosis with pop beads since there was some confusion, especially on keeping track of the genes. So to give you an idea, I have a cell here. It has already replicated its DNA during interphase. So most of the time our cells, the chromosomes really will look more like this. Because there's no reason to carry around two identical copies of every molecule of DNA. It would be like carrying around two copies of your textbook. You're not going to gain any information by having two copies, and it's just going to be really bulky. Now notice on this chromosome, which is blue, representing that it's the paternal chromosome, this has gene A on it. So let's say this maybe codes for something like your thumb type, whether it's straight or a hitchhiker. So this has gene A. And then this is the maternal chromosome, similar size and shape, and it also carries that gene A for the thumb shape, but it has a different allele. This one's showing a large A, this one is showing a small a. So same gene, different alleles, different version of those genes. And then I have another type of chromosome here that's a little bit shorter. This chromosome is carrying gene B, so let's say that is coding for whether my earlobes are connected or if they're the floppy kind. So this paternal chromosome has the little b for that, and this maternal chromosome has that same gene, I'm calling gene B for earlobes, but it has the other allele, the big B here. So during interphase, the chromosomes are going to replicate, so DNA replication will take place, so that now each chromosome is made up of two identical sister chromatids. But we still just have 4 chromosomes. Each of these units is called a chromosome. So at the start of meiosis, during prophase 1, the homologous chromosomes are going to pair up. So homologous chromosomes are the same size and shape, and they carry the same genes. So these two are homologous, and they both carry that gene I'm calling A for thumb shape. These two are homologous. They're the same size and shape, and they're both carrying the gene B for earlobes. Now, one thing that happens during prophase 1 is crossing over. So little pieces, sections of the chromatids that make up these chromosomes can actually cross over and switch places. So they actually exchange a bit of genetic information between the paternal chromosome and the maternal chromosome. And let's say up here, it happens between these two sections. So I'll just pop those over. All right, so the next phase is metaphase 1. And so what's going to happen is these kind of groups of chromosomes are going to line up along the center of this cell. Now the orientation of how these chromosomes line up, whether it's this way or this way or this way, that's completely random, and that's really consistent with the idea, that principle of random assortment. So they're going to line up with their homologous chromosome during metaphase 1, and then during anaphase 1 they're going to split up. So they're going to say goodbye to their homologous pair and start moving to opposite ends of the cell. This is anaphase 1. At telophase 1, they've made it to the other side, and this big cell is going to split into 2. So this is the end of metaphase 1, the first division of the cell. Notice I started with one cell that had 4 chromosomes, and I'm ending up with 2 cells that only have 2 chromosomes. So they have 1 chromosome of each type. They have a long one and they have a short one. And we've gone from a diploid cell having 2 copies of every chromosome to 2 haploid cells which only have 1 copy of every chromosome. So now the cell is ready to go through the second division, or meiosis 2. So the same thing's going to be happening on both sides, so I'll model those separately. The first thing, prophase 2, not much really is going to happen here. During metaphase 2, the individual chromosomes now are going to line up along the center of that cell. So they no longer have homologous pairs to line up with, they're just lining up individually. And then during anaphase, the sister chromatids are going to get pulled apart. So during anaphase, they're going to move to opposite sides of the cell. At telophase, they're at the other side. A membrane's going to regroup around the nucleus, and the cell will split again. So I'll go ahead and show that on the other cell. So it's in prophase 2 right now. During metaphase 2, the individual chromosomes line up. During anaphase 2, those sister chromatids split and go to opposite sides. And during telophase 2, the membranes regrow and the cell divides into 2. So at the end of meiosis 2, we now have 4 cells. Each cell has 2 chromosomes in there, one of each type. And these would represent now our gametes, such as eggs or sperm. So I want to take a little bit closer look at the genes that we ended up with in each cell. So notice each cell has 1 copy of the A gene for thumb shape and 1 copy of the B gene for earlobe shape. So in this cell, we ended up with a big A and a big B. And I'll go ahead and write that. These are the alleles in this, let's call this an egg. So big A, big B. In this cell, we ended up with a little b and a little a. So this one has little a, little b. In this cell over here, we ended up with a little b and a big A. And in this cell over here, we ended up with a big B and a little a. So just by chance, we ended up with all possible combinations of the alleles for the 2 different genes.